Myself Jagadish Karo, lecturer in zoology, KB DAV College, Niragarpur. This time I am going to discuss about regulation of cell cycle. Cell cycle includes sequence of phases through which a cell passes from one division to the next. The sequence of phases include G1, S and G2. During these phases, the cells grow, DNA replicate and cells divide. The processes are highly precise and carefully regulated. The careful regulation of the cell cycle ensures DNA of a dividing cell is copied properly. Any error in DNA are repaired. Each daughter cell receives a full set of chromosomes. The cell cycle is genetically controlled. The events are genetically controlled and highly conserved through evolution. Any mistakes during DNA replication or distribution of chromosome can lead to mutations. So, there are internal control mechanisms or checkpoints to rectify mistakes and regulate the process of cell cycle. When errors are detected, cells can be stopped division until conditions are favorable or errors are corrected. The internal checkpoints. There are three internal checkpoints. These are G1 and S checkpoint, G2 and M checkpoint and M checkpoint. The G1S checkpoint found at the end of G1 phases. This checkpoint decides whether the cell will be divided or not. Similarly, the G2M checkpoint is found at the end of the G2 phase, and during this G2 phase, this checkpoint determines if DNA has been copied properly. The M checkpoint, or known as spindle checkpoint, it occurs during metaphase. It ensures chromosomes are aligned at mitotic plate. These checkpoints detect defect in cell growth, synthesis of cellular materials, or any damage is found in the DNA. If defect is found, cells do not replicate and divide and destroyed by apoptosis. The apoptosis is the mechanism of a programmed cell death. That means it leads to cell death. So, there are control mechanism in the cell cycle. The control mechanism is found to be same in yeast, plants and animals. This is the picture of cell cycle. Here the phases of the cell cycle G1, S and G2. Then The checkpoints are G1 checkpoint. This checkpoint checks nutrients, growth factor of the cell, and any damage is found in the DNA. If defect is found, then the cell passes through the G0 phase, that is the resting stage, and cells do not divide. Then the second checkpoint is the G2 checkpoint. This checkpoint checks cell size and DNA replication. That means it checks the proper growth of the cell and proper replication of the DNA. If cell grows properly and DNA replication occurs in a right way, then cell passes through the G2 phase and enters the mitotic phase, that is the M phase. And in the mitotic phase there is the checkpoint which is known as M checkpoint, or that is metaphase checkpoint. This checkpoint checks for the chromosome spindle attachment. That means each and every chromosome is attached to the spindle fiber properly. If everything is found to be correct, then cell divide and new daughter cells are produced. Let us discuss in detail. The first checkpoint, that is G1 and S checkpoint, it is the primary checkpoint or key checkpoint, and this checkpoint is known as start in yeast, restriction or R point in animal. And most of the cells passing through the G1S checkpoint end up. That means they do not pass to the next stage. This checkpoint accesses extracellular growth factors, which is known as mitogens, and intracellular nutritional state. That means whether proper nutrition is available to the cell or not. This checkpoint determines whether conditions are favorable for DNA replication to occur in S phase. The G1S checkpoint checks damage to the genomic DNA. Starvation and a lack of mitogens can stop the cell cycle or put a halt in the cell cycle. If cell does not meet requirements, it passes to an arrest stage or pause state or quiescent stage or G0 phase in G1 phase. That means the cell do not pass to the next phase of the cell cycle. Generally, a cell attempts to repair the DNA damage. If unable to repair, the cell undergo apoptosis. That means programmed cell death. The second checkpoint is the M checkpoint, G2M checkpoint. This checkpoint prevents entry to mitotic phase if conditions are not met. This checkpoint ensures all chromosomes have been replicated and DNA is not damaged. If defect is found, cell cycle is arrested and cells attempt to repair the damage or undergo apoptosis if damaged severely. The last checkpoint is the M checkpoint or metaphase checkpoint, or known as spindle checkpoint. This checkpoint operates at the beginning of the anaphase. This checkpoint checks that all sister chromatids are attached to the spindle fiber or not. Cell cycle does not proceed until kinetochore of each pair of sister chromatids are firmly attached to spindle fibers. There are regulator molecules which regulate the cell cycle. The process through checkpoints is controlled by specific protein kinase that take part in phosphorylation and dephosphorylation. Regulatory proteins, these are known as cyclins, are stimulators. The cyclins are controlled by activating specific cyclin dependent kinase or mitosis promoting factors. There are two types of regulators. These are positive regulators. These promote progress of cell to the next phase, which is known as positive regulation. Example of positive regulators are cyclins and cyclin dependent kinase. Cyclins control progression of cell cycle by activating cyclin dependent kinase. Cyclins regulate cell cycle only when they are tightly bound to cyclin dependent kinases. To be fully active, cyclin dependent kinase and cyclin complex must be phosphorylated. There are also negative regulators. These regulators stop cell cycle and the process is known as negative regulation. Example of negative regulators are retinoblastoma protein and p53 proteins. Retinoblastoma are tumor suppressor proteins, prevent tumor growth by inhibiting cell division and monitors cell size. Retinoblastoma restricts progress from G1 to the S phase. p53 is a tumor suppressor protein produced by the activation of p53 gene, which is a tumor suppressor gene. If DNA damage is detected, p53 production increases. p53 stops progression of cell cycle and starts repairing DNA. If DNA repair fails, DNA no longer replicate and cell undergoes apoptosis. Failure of a single regulatory molecule has no effect on cell cycle, as more than one mechanism controls the progress of the cell cycle or the events in the cell cycle. Friends, neoplasia is a disease in which uncontrolled cell division takes place. If a mutation occurs in p53 gene, defective p53 protein is produced, and this defective p53 protein can lead to rapid uncontrolled multiplication of cell, which is known as neoplasia. Let us discuss length of the cell cycle. In human beings, in the early embryonic cell, the length is few hours. This duration is 2 to 5 days in case of epithelial cell. There are certain cells like cortical neurons and cardiac muscles. These cells entirely spent in G0 stage. Thank you. This much for today.